Hello, welcome back to COG 266, Brains, Minds, and Consciousness. Last time we covered some of the features of vision, of how we can transform light even before it starts to leave our eyes. We start transforming the information present in light into better forms that we can make more sense of eventually. Sense is a pun, it will happen a lot. So we'll recap that, but also we're going to continue upward into the brain to see how this light information gets combined further in the visual system. So let's recap. We discussed the idea of ganglion cells, which are able to look at a variety of locations. So sort of they span multiple rods and cones in the eyes on the retina, and they're able to have this interesting response pattern with these center surround cells, where they have this interesting pattern of activation. Light that is present in the center is positive for the cell's activation, but light that is near the edges is negative for the cell's activation. So light present in the center leads to more firing, whereas light present on the edges leads to less firing or no firing. It inhibits, right? Which gives it some interesting properties. So when there's light at the center of a center surround ganglion cell, hey, it's lit up, that makes sense. When there's light on the edges, it's darker, so that the firing pattern is off. And when there's light nowhere, it's sort of this medium pattern. When the light is gone from the cell's detectable range, it sort of fires at this middle rate. This leads to some interesting properties with respect to being able to detect edges. So when there is light, when the whole ganglion cell is covered in light, has sort of a neutral pattern of activation, because the surround inhibition and the center excitement are sort of canceling each other out. But as an edge crosses over and we start to get this lack of light in the surround, but still have light in the center, the cell gets excited, quite excited. A little less excited when we start to bleed into the center, but still that it's still more powerful than when it was all one. And then eventually we get to this sort of negative firing where again, when the edge is here, that the edge is present there, and it has this detection of the edge. So this ganglion cell is able to, in sort of one shot before light even leaves the eye, get most excited when an edge is present. It also detects light, which is just fine, but this is like light in a general sense of, oh, is it brighter or darker? But it also has this curve, this pattern to it. So if we recall, these ganglion cells are overlapping one another. They're kind of all over the place, which leads to another level of abstraction that we have. We're able to say, given a bunch of ganglion cells together, we can sort of think of them as a pattern in the world, and given these properties of edge detection, you can also imagine sets of cells that are lined up to detect only particular patterns of activation across these ganglia. For example, when there is an edge passing through these three ganglia and not any of the others, that might light up the first set of abstracted detectors, whereas the vertical pattern might light up the second, third, fourth. Now we have the idea of orientation detection. So we have sets of as we keep going, these neurons all bind up to other neurons. They excite each other and inhibit each other the same way that the receptors did the ganglion cell. And now we have a more abstract concept. So when the pink bar is the only one there, that's the right hand diagonal. How do I say this? The 4/, right? Angle detectors are lit up, because those ones are bound to say, hey, when these angles when these three ganglia are active and most excited, I want to be excited about that and inhibit all the rest. Whereas when the pattern changes to the others, you get different things. So when just that pink bar is there, this angle is detected. Now, color. If we recall, we have an eyeball, and we have light as a wave that can come into the eyeball and activate our receptors. There are two properties of light that we find interesting. There's the amplitude and the wavelength. These are the physical properties, right? We know them as different things because we translate them as they come into our eyes and they hit our three receptors, right? So we have three different color receptors typically, and they are tuned to react to different wavelengths. So a certain wavelength will activate the blue receptor much more than it might activate the green and red. And remember, they overlap again. So when we have 445 nanometer light, that means the blue is quite active and the green is only very slightly active. So you take a vertical slice of it, and the red is almost not active at all when that light hits there. So these get abstracted up into these opponent processes of how much red versus how much green is present, how much blue versus how much yellow is present, and how much dark versus how much light is present. And then we get things like hue and brightness. And just a reminder that it's these three, the fact that we have these three types of cones that allows us to detect the full range of normal color vision. But a lot of people don't have all three types of cones, and they end up with different types of color blindness, right? And a reminder that we are all colorblind to an extent because mantis shrimp can see way more colors than any of us will ever dream of. So I guess we're screwed. Oh well. But that's the idea. So we take the physical properties of light and we convert them to these sort of psychologically meaningful properties of the hue, the color palette, the brightness of the color that I'm seeing. Also saturation, which we haven't talked about, but is another sort of distinct process. So that's the recap of where we've been with light so far. Now, let's talk about how this information moves past the eye, upward and onward into the brain. So, visual cortex. When visual information leaves the eye, it heads into the visual cortex. The V1 is what it's known as typically, the early visual cortex. And it begins processing for basic spatial information, things like we've already talked about with orientation detectors, right? So again, follow along. We have our nice cross-section brain friend here. The optic nerve sends information on, and once again, it's not an anatomy course, so these are just the names of things as they're written. So lateral geniculate cortex, and it all hits the back in the occipital lobe. We've talked a little bit about how the occipital lobe is where the visual information happens, and it stays a little bit sort of spatially connected. So to recap that, a little bit more of a look at how these things are physically laid out. In your eye, on your retina, you have these bundles of cells. There are the photoreceptors, there's the second order cells that are sort of the ones that catch, the second messenger thing. And then the ganglion cells, which say, hey, what's the overlapping patterns, where are the edges, that kind of stuff. And in early research, we found that in the optic nerve, there were some patterns that responded most to light being present. There were some nerves that would be more active when things were dark, and there were some that would only really be active when something changed. All three of those, you can imagine, a pretty important type of information. I need to know kind of how dark it is, how bright it is, and I also need to know when something changes, right? So, on the way in, we start to see some simple combinations. So there are both simple and complex cortical cells. Simple cells just take information and concatenate it in a sort of standard way. So what do I mean by concatenate? Well, you might have, say in this example here, you might have 5 separate cells that are all doing this sort of the center surround thing, right? You might have a cell that integrates all of those and says, as long as there is a long bar and then negativity around the edges, it's not, what it's doing is saying, I'm going to simplify this signal, these things that are all right next to each other, into a single pattern. Whereas complex cells might take information and do something a little bit more complicated to change it to a sort of new form, right? So not just kind of staple them together, the signals all together into one blob, but recombine them. So again, looking more carefully at it, the simple cells are going to take a bunch of other cells' information and just say, I'm going to respond when this pattern is more or less the same exact pattern, but kind of simplify it into a single signal. Whereas a complex cell is going to take, say, you have these simple cells, and they say things like, so these three different cells have different patterns of activation. So some of them might be high, some of them might be low, and you recombine them into a new sort of summation. Something like the opponent process, as we talked about, right? So recombining all of the patterns of green light and red light that we're getting off of the retina, and then putting them into a sort of an opposed single value of how much red versus green, right? So many cells get transformed into one signal. So in the same way, say these are all simple cells that are next to each other in the receptive field, and they are all, literally physically next to each other. You might have this recombine in a way that's helping detect, okay, how far up the visual field is a given light, right, or a given amount of light. That can be, so you can recombine, so say the top receptive field on the left there is lit up, and the other two are dark. We can recombine that into a high firing rate for the fact that this thing, the light is high up in the field. And the opposite pattern, if the top two are dark and the bottom one is lit up, then that could be combined into a very low firing pattern in that, the thing is low in the receptive field. Now we've translated all these different firing rates of where light exists in physical space and combined them into a single representation of how high up or down is the light, I can tell you with a single firing rate. That is it's a simple form of complexity. I know that sounds like a paradox, but it's complexity in that it's not just straightforward mapping one thing onto the others. It's sort of doing this combined, each thing can influence each other and all these things. So that's the idea of a complex cortical neuron. We can take information in one form and transform it into a simpler but more informative form. Now, this idea carries forward a bit into the concept of tuning curves. So, similar to vision, right? We know that different cones are sort of tuned to particular color wavelengths. All other neurons can be tuned to respond mostly to particular stimuli. So, interconnected adjacent cells that wire up to detect a variety of angles, for example. So, on the bottom left, we have the color tuning curves for the cones in your eyes. On the bottom right, we have the tuning curve for a vertical orientation detector. It responds most at the middle, right? So there's the most response in the middle when the vertical detector is present, and it has some kind of falling off of response. As an orientation that is present in vision becomes a little more horizontal, this neuron will taper off in how much it responds. Now, the real beauty of this, well, I mean, there's plenty of beauty in it, but the tricky part is that now you can overlap several of these neurons and you can complete the gradient to get sensitivity to all orientations. You may only have neurons tuned to, as on the last slide, there's about 1 neuron for each 15 degrees of orientation. But we are not only sensitive to those degrees. Just like how we can combine the sensitivity of the 3 different kinds of light to experience the full visual range, we can combine the sensitivity of different orientation detector neurons to get the full experience of orientation. Now, to see, it's kind of cool. These things kind of stay connected as they enter the cortex. So here is a sample. The pattern of various tuned neurons, they're colored by orientation, right? So the purple are slightly angled to 4/. The green are sort of horizontal, the red are vertical, and so on. That is a sample of the neurons that are detecting for a given little area, the ones that are responding to vertical orientations and the ones that are responding to horizontal orientations and et cetera. This is not a visual field so much, but it is the orientations of the visual field. This is a few layers up, right? Just an interesting little bit. We'll touch on this again in a minute. Speaking of touch, we can see similar patterns of angle detectors in touch, right? So we have, in the same way that in vision, a certain set of neurons being active might cause a particular angle detector to trigger. Exactly the same on your palm. If you do push your fingernail into your palm one direction, it feels one way. Push it into it a different direction, it feels a different way. You can also don't push too hard. The exact same thing. There are tuning curves designed to capture those relationships. So this brings us to an interesting concept that is key to all of sort of understanding brain activity stuff. Self-similarity. So we're seeing how the cone's tuning curves use this discrete set of sensitivity profiles, right? There's 3 colors, red, green, and blue, to capture this wide range of color experiences and send them onto the brain. That's the same way that orientation neurons use that exact same process to capture a different type of information from the light in the world, right? So they are getting orientation in a similar way that we are getting color. This is self-similarity, and it's the idea that the same process and structure applied in different places can achieve very different and increasingly potentially complex things. So to carry this forward, you can also use this same concept of tuning curves to combine information from this step, right, from orientations, up into something more complex than those, right? So I may have neurons that are sensitive to particular orientations of edges in the world, but I can also have neurons that are sensitive and tuned to particular combinations of orientations, right? So there are such things as facial tuning curves. So light becomes differences, becomes edges, becomes orientations, becomes shapes, sets of shapes, and finally faces. So we have the response from non-face, face to non-face. We can watch macaques. We can watch the patterns of activation in their brains as we present them with facial stimuli. Let's see. So a guide to reading these slides. In the box at the top, you'll see when there is no stimulus, it's white. When the stimulus is present, it's this sort of teal gray thing. And then finally, when there's no stimulus again, it's back to white. And time moves rightward like an arrow. So macaque face detection neurons. For frontal, so when you present a macaque with this image, we get great activation in that set of neurons. They say, oh, there's a face. I like face. Face is good for this neuron. This neuron is tuned to this combination of features. If we scramble it, neuron unhappy, neuron inhibited. The wrong combination of features is present. Even though they're all still there, the eyes are there, but they're in the wrong places, right? Part of the mouth has moved, the nostril has moved, we have broken the pattern, right? Now, to show an interesting difference here, if I just strip out some part of the face but leave the overall pattern intact, well, we get a little bit of activation back. If I restore the face, we get even more activation back. So these pattern detectors, very much like light, you got to think of this as the tuning curves allow for this sort of soft failure of, well, if the pattern's not complete, then I can still, piece together what's going on. If there's a bunch of other features present, like in the scrambled face, that kind of screw up my pattern detection, then that disrupts the whole thing. It's hard to pin down exactly how that transformation happens, but start here, right? Start here with the knowledge that a scrambled face will not activate these neurons at all, whereas a partially incomplete face will. A human face actually does trigger it. It has all the right features in the right places, but they have sort of they're a little off from what the macaque is trained to do, right? Now, as a control, what about a hand? Well, a hand has nothing compared of all these faces. And so, as expected, there's almost no activation, no real activation. So, that's macaques looking at frontal face panels. It also works for the profile. So you have neurons, and this is a little bit even more in the tuning curve life. Let's look at a set of stimuli that are presented along this profile curve. So a front-facing macaque, a slightly turned macaque, a more turned macaque, a fully sideways macaque, and an almost sort of looking away from us macaque, and a toilet brush, which has the same rough overall features, at least on the outside edge of the macaque, with sort of the fuzzy outside line and sort of a bright inner space. Let's see what happens. So the profile neurons, when the macaque is facing forward, they don't fire, right? They're not trained for that. They don't care about that. The macaque's slightly turned, not much going on. Similarly, not a whole lot of activation. As it continues to turn, though, it lights up. And potentially even more based on this particular pattern. So we see the curve, right? We're seeing the tuning curve writ large. It's showing that this is a set of neurons that are excited most by profile. This is great. This is useful for an animal that lives in a social setting to know, oh, this person is this not person, but this other of my kin is looking away. Maybe they're looking at something important. Maybe I should also look at something important, right? Also, the toilet brush doesn't have any activation at all. So that is demonstrating how we can sort of we see activation for face patterns in macaques, and there's that it also works for humans. So why the curve, right? Why are we doing this curve? I've already more or less, I think you may already be getting this, but it's good to be explicit about it. If neurons were just hyper-specific in what they were able to respond to, they wouldn't be able to be combined over, things that deviated from that at all, right? So if I only had vertical and 45 degrees orientation detectors, and I had a very sharply specific response and not a sort of broad tuning curve, then how would I detect either of the orientations present over here, right? The sort of 10 degree graded, the 35 degree graded, etc. You wouldn't be able to, right? But more than that, pardon. It's not just being able to detect and not being able to detect. It's also about the precision, and we'll walk through that. So, how accurate are these detectors, right? Clearly, we have to have some kind of limit in terms of how sensitive our neurons and receptors are, right? There's only so many of them in the eye. They take up a physical amount of space. They can only be firing or not, right? So we can't see infinitesimally small. We can't see individual atoms, right? We can only see a certain kind of pattern. But the fact is that we can combine activity from nearby sources to achieve what's known as hyperacuity. Hyperacuity is the ability for us to detect patterns that they seem like they're beyond the scope of the neurons on their own. Let's do an example. So, here's the question. Are these dots horizontally aligned? Like if I laid a leveling tool on top of them, would it measure that they were flat? It's kind of hard to say. What about these ones? Probably feels more right. And these ones, a little easier, right? Dots don't give us a lot of information to work with. You can sort of tell, but it's a coin flip, right? But if I offset two lines the exact same amount, you know that immediately. Those are not perfectly aligned. These ones are, and these ones, again, even further, are not. The question is, why, with the lines, is it easier to detect displacement? Displacement is interesting. So back to our ganglia friends. As we saw in that recap, ganglia are a little bit graded depending on how close to the center the activation is, or rather the light activating the edge is. So in the bottom left here, that means that when the edge, when the pattern is close to the center, it's strong, and as it approaches the edge, it's a little weaker, right? Or the edge of the receptive field of the ganglion, right? So when it's kind of at the outskirts, it's not a very strong activation. When it's closest to the center, and then when it's at the very center, it's a very strong activation. We also know that they're overlapping. So we have lots of ganglia near each other, or ganglion receptive fields near each other, overlapping with each other a bit, so that these things, when there is a continuous line, is going to activate a whole bunch of ganglia together. Meaning that if we have some neurons that are kind of set aside to try and detect differences, the overlapping and graded responses give this really detailed pattern so that we can really tell when sets of things that are long and continuous are separate, right? So as long as the stimuli are spatially extended and they are displaced slightly, it produces a wildly different pattern of activation. The reason that dots don't is because they are not activating enough nearby cells. And again, the idea is that we can tune neurons to these different patterns. When there's not enough information, there's nothing really to tune to. So it's harder to produce that same pattern with dots than it is to do with lines. That's displacement hyperacuity. Back again, so in general, tuning curve hyperacuity, this is the same kind of idea as vision. I just wanted to lay it out again. So the fact that neurons, in a general sense, can integrate signals from lots of disparate sources means they can be more sensitive than just they themselves are, right? Because we can combine, and we can always move up a step and recombine the patterns of several related neurons into a new signal, and then recombine those patterns into a new signal again. So your color receptors, again, leads to your experience of color. Let's run through a few examples. Oh, let me get all of these up. So your red, green, and blue responses and your experience of color. So blue, you're going to have a whole lot of blue, a little bit of green, and almost no red. You combine those to get this sort of dark blue. We do the same with this sort of teal. We get a little more red. We get plenty of both green and blue. In fact, an equal amount of both. activations, right? So those cones are active more and red is active a little less. Combine those down into this teal color. We get our greens, we get our yellows, we get our reds. This is a very important image, I think, for you to understand. The idea that patterns of activation can be combined into something more than just what they were, right? So this set of patterns of activation are key to producing the output values of sort of this color experience. This is the process of abstraction, which will come up again momentarily. So, to recap, cortical vision. There are neurons in the cortex that are tuned to respond to different combinations of these basic stimuli. They increase in complexity from simple center surround to orientations to shapes to faces, and then ultimately the fact that these all have graded patterns, they have graded responses, enable us to detect things that we don't have exact tuning for. There's of course some inaccuracy, but there's also hyperacuity. We can get extra precision by combining over things in groups. So let's move on to vision modularity. So after basic visual processing, we start to send the information on to other parts of the brain, right? So information will get increasingly transformed and abstracted into these more complex concepts, we can call them, or simpler patterns that are more meaningful. They combine more and more information into a single, tuning curve. But they can also be sent in parallel to different areas and be processed in different ways. So that's the general idea of modularity. Now, before we get to the modules, some of these modules, let's talk about the path upward. So we've already looked at a little bit of this, but the path upward from the pattern of activation of the light carries upward into the brain in these what are called hypercolumns. So again, we've already looked at light becomes edges, becomes orientations, and we see that it carries forward in the same way upward, right? And gets combined more and more once we get to the visual cortexes. We'll talk about some of those in a minute. An interesting feature of these hypercolumns is that for a while they stay pretty faithful to the sort of the organization of the retina, right? So this little region that we see down here highlighted of these orientation detectors is roughly spatially accurate, right? It's still connected to, that's something that's in the sort of bottom left part of the visual field. That's not the way it stays forever. The further up we go, the more sort of diffuse and less related to the exact physical location they get. But so there are these large areas of these overlapping orientation detectors. So that carries upward into the brain. Now, once we get there into the brain, we hit a whole bunch of different modules of vision. So these are different visual pathways, different functions of vision. Then they create different outcomes that then other parts of the brain can sample from. Your frontal cortex needs something to work with. And so on the way up, the light patterns get changed and chopped and moved and transformed into things that are useful, like recognizing what a thing is. So let's chase down that particular set of modules, the what pathway. So also, again, as usual, this is to demonstrate that there are a bunch and they're all interconnected. This is one theoretical model of this. It's not a perfect representation. You're not on the hook to memorize what that is. But the idea is that visual modules all throughout the occipital lobe are doing different work, right? They are moving things around in different ways. So what's the what pathway? This is the interconnected set of littler transformational visual modules. They're going to do different things to information, but pass along to various further systems down the way. So you'll see that the information evolves. So V1 is the first chunk. The primary visual cortex And this is mostly about just catching all those oriented edges, right? V2 is the secondary visual cortex, which is going to represent junctions, intersections, connected oriented edges, say angles, for example. That's the easiest way. From V2, we hit V4, which is more complex shapes, a little bit wider range of possible shapes that they can be tuned to. Then we get to IT posterior, which will detect entire objects over a wide range of locations in the visual field, different orientations of faces. Again, so we're combining these three different raw shapes might be connected to the same idea of an object, right? And then IT interior, so visual information becomes sort of abstract, meaningful. We start to be able to connect that up to semantic memory and catch things like, oh, this is a person, and I know who they are, or oh, that's the London Bridge, or whatever, right? Objects. So finally, from the bottom to top, we have raw edges, we have junctions of edges, we have or oriented edges, right? Junctions of edges, shapes, multiple possible shapes, finally to the identity of the object that we can then bat around in the brain. To look at that a little more cartoonishly, the blue fellow is examining the green fellow. In V1, we get just a pattern of oriented shapes. In V2, we see that we get these junctions, these connected parts. V4 gives us the shape of, oh, that's the circular bit and the leggy part. I'm starting to see that this might be one face of a person, a little circle person. And finally, I might recognize that, oh, that's green. I know green. Right. Does this make sense? This is so the same way that we have tuning curves for orientations, and the ganglion cells can detect edges. We continue up and have tuning curves for joints, tuning curves for shapes, tuning curves all the way up. So just to give you a peek at the full kind of range, maybe not full, but V2, it's not just junctions, it's almost a full suite of emoticons. They have different abilities, so lots and lots of shapes are able to be captured in the V2 area. They're not all active to the same amount, but different areas can handle different bits. So these shapes, you see there's sort of curves, there's boomerang shapes, there's slight curly braces, there's hard right angles, there's carets, there's asterisks, right? V4, on the other hand, you continue up and you get into more sort of whole shapes, weird shapes, because these are they're still just super basic features of more complex objects, right? These are not complete, this is not where we recognize that's a dog. This is where we start to say, there is a circle next to a circle, and some other area of the brain will say that's a snoot, right? So V4, these are the kinds of things that V4 is more active for. Now here's a little nice cutout that shows sort of the kinds of things as they progress up. So V2 might give us a couple of small angles and concentric circles. V4 is going to give us the ability to connect full-on shapes. Posterior IT is sort of recombines those shapes into new forms until we get to anterior IT that's able to sort of extract a little bit more. Oh, this might be the edge of a hammer, that might be somebody's face, that might be a ping pong ball, a target, whatever. And so, again, these are artists rendering, and they're almost always scientific artists renderings, so they're not perfect. But that's the idea. We see the flow upward. Beyond anterior IT, we can get into faces. So you have a part of your brain that beyond even what we've talked about so far for the visual areas, you have an area that it's dedicated explicitly to detecting faces, the fusiform facial area. So here is a raw pattern of light hitting your eyeball, right? We know already, you know already, in fact, proof is in the pudding, this is a face. You know it's a face. You knew immediately it was a face. It's one of the best things, that's one of the things we are best at, is recognizing human faces. How do we get there? So again, low-level features. There's a bunch of possible orientations and shapes, brightnesses, center surrounds, darknesses, gradients, regions on the face, right? Those get combined upward into more meaningful face-like shapes. So little curves, those help us see eyebrows. Little concentric circles, those help us see eyes. Kind of curvy bits for the nose. We have circular bits for lips, and well, not circular, I guess, but we might have circular bits for open mouths and sort of these flat lines for closed mouths. We can get little circles below curves to get nostrils. Those features then all get combined up. Again, think of each square here as one neuron is tuned to that particular pattern. The pattern of light creates the low-level features, right? But then at the mid-level features, those are tuned to features from the low-level features, right? So those neurons are tuned to patterns from low-level features. High-level features are full facial parts. And again, I hope you're following the idea that you can combine parts from the mid-level features to create tuned neurons that are sensitive to these kinds of connections. You know, we have one face here kind of in the top row. That guy has a mustache, whereas the one right next to him does not have a mustache. And those are both different tuning curves. Those are both neurons tuned to a different type of face. Which brings us to the general concept of pattern recognition. Now, these faces are patterns, right? But there's something interesting about what we're capable of doing with patterns. It's kind of tricky to do this if you're an AI researcher in the early days. How do you get something that is invariant, right? So there's lots of possibilities for conflict. How do we differentiate the A and the F? Look at that. They are almost the same, except there is a chunk is missing to create the F. We know those are different. They're different patterns. But how do we know that the top A and the right A, the top right A, are the same, even though they have very little in common? In fact, you see that there's only sort of the bottom right stick is in common between the two. But we know that they are both A's, just like we know the bottom two are both F's. Same with the bottom. All three of those are A's, right? How did we get this? Well, it's the idea that relationships and spatial locations, they're both information. And so neurons can handle both of these cases. So the raw inputs might be wildly different between the A in the upper left and the A in the bottom right, right? But the relationships between those inputs are identical in the case of this sort of just different spatial location, right? Now, it's a little bit more complex when you're dealing with orientation and when you're dealing with, like, you know, the larger versus smaller shapes. But the idea is that the differences between the outsides and the internal bits of these shapes, they maintain something in common, right? The spatial locations are information, but the relative patterns are also information. There's no reason that neurons can't handle both. So in this example here, we have in the upper left, that A, you might have an array of A recognizers, and there's one for each little area, right? And so the fact that an A recognizer is saying, hey, there's an A in the upper right corner, but an A recognizer saying, hey, there's an A in the bottom right corner, those can both be you know, sets of these are stand-ins for more complex combinations of features like we just covered, but this is the idea, right? So some kind of group of neurons can say, oh, there's an A over in this spatial area. So those can all then be abstracted up to the A finder neuron that says, hey, if any of you guys are active, I'll be active. So if anywhere in this visual field that you're kind of covering, I'll be okay with any of those. Now these are laid out for spatial location, but you could also imagine now for different orientations at each spatial location, right? So as long as something is able to detect the pattern, that pattern can vary with where it lives in both space and orientation and all these things. This is how we're able to connect the dots and say, oh, these things, even though they have different surface level specific features, they have a pattern in common that lots of my other neurons can sort of pass up to this pattern detector. Now, again, there's no evidence of literal A recognizers, right? But this is to carry us forward on the idea of abstraction. So the key to all of this is the idea of abstraction. So we've seen, look at this little image on the right, way at the bottom, we have detecting the raw patterns of light. The neurons are going to combine more and more complex stimuli, and they're going to become more and more sensitive to abstract concepts, more information-rich, information-dense concepts. Every time you have a neuron tuned to the outputs of a bunch of other neurons, you're combining more information. That's how we get from edges and lines up to full faces, right? You can have a neuron tuned to one full face. Now, to help with the idea of sort of the invariance of being connected over this space, so the higher areas are sampling over this larger set of abstract signals, such that the lower areas, they might only respond to very specific locations, right, in the visual field, but at the higher levels, they're becoming more and more abstract automatically because they're not connected to the raw physical locations anymore. They become spatially invariant. The information gets subtly transformed the higher up it climbs. That's fascinating to me. That's sort of one of the most powerful concepts here when we're trying to understand how a neuron, which now you get, right? Patterns of activation of a neuron, how light gets into it. Its activation combines with a bunch of other neurons and goes up a layer into neurons that are sensitive to different features of those neurons, into more abstract features, until we get to something like a face detector. Now, this is very cool, right? This is the big idea of how we are going from raw inputs to abstract detections of concepts. But how then do we retrieve meaning? Meaning. What is meaning? So when I see a hammer and I know that I can use it to smash a nail into a thing, that is meaning. But it's not present in the hammer in the world. It's just a thing. It's just a pattern of light. And no matter what I'm doing to it, patterns of activation, up, how do we take these shifting patterns of activation and get these unified, continuous percepts of what things are and how the world is and what's happening around me? So as we increase in complexity, I mentioned this before, it gets harder and harder to study the exact machinations, right? Vision is so well understood because vision is thankfully sort of spatially connected. But as we spread out into the whole brain, it gets really hard to follow where those edges and eye detectors go, right? Some of them might go off to memory, some of them might go off to decision making. They start to spread out all over the place in a way that is computationally really friggin' hard to pin down. So this is the binding problem. This shows up all over cognitive science. Oops, sorry, I banged into the mic arm. This shows up all over cognitive science in that it's kind of always on the tip of your tongue when you say, well, how does that neural activation become, a feature of mind? Well, That's because we can be comfortable with both. So at some point, while we're studying the brain and the activations, and while we keep in our hearts, we know that the brain is the set of neurons activating each other in all these cool ways we've talked about. But at some point, we got to jump the shark and abstract up to mind. And that's finally where we're headed. We are getting to the concept of mind and how it behaves, what are its functions and features, and how does it sort of interface, live on top of the neural substrate that we've covered in such detail. So, next time we'll move on to mind. As always, here's a final thought. Consider how the brain makes use of tuning curves to allow us to respond in a continuous way to patterns in the world. For example, it's not just yes or no to whether a face is present, but rather how much a thing looks like a face. Now consider your own lived experiences. You likely often get a feeling about something along this sliding scale, maybe a sense of uneasiness or excitement. Thinking on one of those cases, what simpler patterns might your neurons be combining to increase or decrease your sense of this complex feeling? All right, thanks. Take care.